Assalamualaikum and a very good day to everyone who are watching this video. My name is Nurizati binti Ghani. I'm a master student from University of Malaya. Today I'm going to present my master project entitled Characterization of multidrug-resistant pathogenic Acinetobacter baumannii by using next generation sequencing approach. For your information, this project is supervised by Professor Chan Kok Gan. As for the introduction, Acinetobacter baumannii has emerged as an important nosocomial pathogen and constitute a major problem in hospital, not only in Malaysia but also worldwide. Acinetobacter baumannii has caused a wide range of clinical complications such as bacteremia, meningitis, respiratory and urinary tract infection, especially in immunocompromised patients. Not only that, multiple studies have reported that this Acinetobacter baumannii has been the causes of the nosocomial outbreak, especially in intensive care unit. Over the decade, these bacteria have evolving until it become multidrug resistant and have compromised the effectiveness of the treatment option, which can result in antibiotic failure. Carbapenem is one of the antibiotics were once considered to be one of the last resort, but rapid worldwide dissemination of multidrug-resistant bacteria has made them the first or the only option left for many infections, and one of them is the carbapenem-resistant Acinetobacter baumannii, which have been observed worldwide, mainly driven by the spread of the multidrug-resistant clonal lineage. Because of this, WHO has considered this carbapenem-resistant Acinetobacter baumannii as critical priority pathogen for the research and development of a new antibiotics due to their limited therapeutic option left. Unfortunately, in Malaysia, this detailed information of the antimicrobial resistance surveillance is still lacking, especially in nosocomial pathogen. Therefore, further research is actually required, which could help to provide a better outbreak control. The objective of this study is firstly to identify the multidrug-resistant pathogenic bacteria strain 930. Second is to determine the antimicrobial susceptibility profile of this strain. And thirdly, to analyze the antibiotic resistant genes in strain 930 by using two approaches, which are by using bioinformatics as well as the genomic approach. For the first objective, we identified the strain 930 by molecular method, which was 16S rRNA sequencing. We have collected the sample from the local laboratory diagnostic collection, which isolated from the patient. We cultured the strain in the Luria bertani medium at 37 degrees Celsius and extracted the DNA by using Kaizen kit following the manufacturer's instruction. After that, we amplified the 16S rRNA gene by using PCR, extracted and purified the band before sending for 16S rRNA sequencing. After that, the sequence were aligned and trimmed. Sequence similarity were performed by using EasyTaxon database as well as NCBI database. The phylogenetic tree was performed by using Mega software version 7. For the second objective, which is to determine the antibiotic susceptibility profile of Acinetobacter baumannii strain 930 by using this division assay. So, an overnight cultures colonies of the strain were used to prepare a standard inoculum by using a sterile PBS, and then the turbidity of the inoculum were adjusted and compared to the 0.5 mcfarland standard solutions. After that, the culture was spread on Muller-Hilton agar within 15 minutes after the turbidity is adjusted. A sterile cotton swab was dipped into the inoculum and removed the excess fluid, and then the swab was streaked all over the surface of the agar, and the plate were allowed to air dry before the application of the antibiotic disc. After that, the antibiotic discs were dispensed aseptically onto the inoculated MHA plates. The discs were gently pressed onto the surface of the agar to ensure the complete contact of the disc with the agar by using a sterile forceps. The plate were incubated overnight at 37 degrees Celsius, and the zone of inhibition were measured and interpreted according to the CLSI guideline. For the third objective, which is resistome analysis on multidrug-resistant A. baumannii strain 930 by using genomic and bioinformatic approach. After the strain were confirmed to be multidrug resistant, the strain was subjected to whole genome sequencing. the DNA will be cut into a short fragment which later were amplified by PCR. The adapter were ligated and the DNA library will be loaded into the Illumina My6. The sequence were assembled and the antibiotic resistant gene analysis were performed. The assembled genome were used to design the primer for the gene of interest in this study. For the genomic approach, we have amplified the gene of interest, which was PLA-OZA23 by using PCR and ligated onto the topo vector by using topo cloning kit. The reaction was set up as in the table and gently mixed and incubated for 5 to 30 minutes at room temperature to obtain the desired construct. The transformation experiment were conducted to confirm the gene functionality. The top 10 E. coli competitor cells were thawed on ice before mixing with the desired construct. The cells were incubated on ice for 5 to 30 minutes and then the cells were heat shocked at 42 degrees Celsius for 30 seconds and immediately transferred into the ice. After that, 700 microliters of Luria Britannia broth were added and incubated horizontally at 200 rpm at 37 degrees Celsius for 1 hour. After that, the cell was centrifuged for 3 minutes and the supernatant were discarded. 100 microliters from each transformation was spread onto the pre-warmed selective plate containing the ampicillin and incubated overnight at 37 degrees Celsius. After that, 10 white colony were chosen and the gene was screened by using colony PCR. The transformant were further plated onto the plate containing the desired antibiotic, which was meropenem. Next, moving on to the result and discussions. For the first objective, the 16S rRNA gene were amplified as shown in the figure 1 and sent for Sanger sequencing. Lane 1 indicate 1 kb DNA ladder. Lane 4 is the strain 930 which used in this study, and lane 6 is a negative control. Multiple alignments and sequence similarities of the 16S rRNA gene were determined with all existing sequence of type strain of Acinetobacter retrieved from the EasyTaxon database. From the EasyTaxon database, strain 930 had shown 99.93% pairwise similarity with type strain Acinetobacter baumannii ATCC 19606. Not only that, the sequence similarity also was performed against NCBI database and had confirmed the strain 930 as Acinetobacter baumannii with 100% identity similarities. Figure 1 shows the phylogenetic tree analysis of 16S rRNA gene for Acinetobacter strain 930. The phylogenetic relationship between sequence of 16S RNA was estimated by neighbor joining method. After the construction of the phylogenetic tree, bootstrap replicates were performed to estimate their node reliabilities. Bootstrap value was set at 1000 repetition to determine reliability of each phylogeny. Acinetobacter eworfi and CTC 5866 serve as the outgroup. Strain 9380 is mostly closely related to Acinetobacter baumannii because they were placed on the same clade as the reference strains Acinetobacter baumannii ATCC 19606. Table 1.3 show the taxonomy of the Acinetobacter baumannii strain 930. Therefore, strain 930 was identified with high confidence level as Acinetobacter baumannii. Next, moving on to the antimicrobial susceptibility profile of multidrug-resistant Acinetobacter baumannii strain 930. The antimicrobial agent chosen in this AST test are based on the treatment used in treating A. baumannii infection as well as from different classes of antibiotics. The first line treatment for the A. baumannii infection is broad-spectrum cephalosporins such as cefepime as well as ceftazidime. Carbapenems and beta-lactamase inhibitor were used if the strain were resistant to cephalosporin, and in a setting where the strain were resistant to above agents, polymyxins such as cholesterol and tetracycline such as minocycline were used. The antibiotic highlighted in the yellow color are the antibiotic used to treat the Acinetobacter baumannii infection. From the table, it showed that the strain 930 already resistant to all antibiotic listed. The table showed the microbial susceptibility profiles of the strain 930 against quinolones, tetracycline, aminoglycoside, and folate inhibitor. It can be concluded that Acinetobacter baumannii were resistant to more than 3 groups of the antimicrobial agents, including aminoglycoside, penicillin, cephalosporin, quinolone, beta-lactamase inhibitor, as well as carbapenems. The strain was susceptible to sulfametazazole trimetophorin, and minocycline and doxycycline remain effective against Acinetobacter baumannii strain 930. Figure 2.0 shows the AST tests that have been conducted. It can be seen that a high rate of carbapenem resistance has been explored in several similar studies. Firstly, in a study conducted by H. Masomi in 2021, revealed that 93.84% Acinetobacter baumannii were identified to be multidrug resistant. Second study conducted by the Hazir Kamal had revealed that 95% of Acinetobacter baumannii were identified as multidrug resistant with high resistance towards ceftazidime, imipenem, meropenem, and ampicillin sulbactams. And similar studies were conducted and had found that 100% of the isolate were resistant to ceftazidime and cefepime, with high resistance toward ampicillin sulbactam, imipenem, and meropenem. From the finding, Acinetobacter baumannii strain 930 are confirmed as multidrug resistant as well as carbapenem resistant. For the third objective, whole genome sequencing were conducted. The sequence obtained will undergo quality checking and assembled by commercial software. Gene prediction and annotation were performed by using PROCA, Prodigal, as well as RAST software. The antibiotic-resistant gene analysis were carried out by commercial software, which are STAR AMR, PlusFlow, MobRecon, as well as Abricat software. The table 3.0 showed the genome statistic for complete genome sequence of Acinetobacter baumannii strain 930. It have GC content 38.8% and 3,838 coding sequences. Figure 3.0 shows the subsystem category distribution statistic for complete genome of Acinetobacter baumannii strain 930, which generated from the RAST software. And as you can see in the table 3.1, the highlighted row is the BLA-OXA23 gene, which were predicted phenotype for carbapenem resistant in Acinetobacter baumannii strain 930. Figure 3.1 showed that the agarose gel of electrophoresis of PCR product for BLA-OXA23 gene amplifications. This showed that the strain 930 Acinetobacter baumannii had harbored the BLA-OXA23 genes, which the lane 1 indicate that 1 kb later. Lane 4 is indicate the strain 930 used in the study, and lane 6 is a negative control. Multiple studies have reported the same findings, where the study by Hamidian and Negro in 2019 found that in Acinetobacter baumannii, carbapenem resistance is associated with the BLA-OXA23 genes. Not only that, a study by the Hassan et al. In 2021 found that the BLA-OXA was the most common detected gene with 77.7%, and the insertion sequence ABA1 detected in 10% of the BLA-OXA23 positive isolates. Another study also revealed that this BLA-OXA23 gene can be chromosomal or plasmid mediated, or can be chromosomal and plasmid mediated. A study by the Wang et al. Have revealed that the BLA-OXA gene can co-occurrence in both plasmid and chromosome, which were believed to increase the fitness in the carbapenem-resistant Acinetobacter baumannii. Figure 3.2 showed that the transformant were plated onto the Luria britanniae agar containing ampicillin and Adscal. Meanwhile, figure 3.3 showed that the gel picture of the transformant with the gene insert of BLA-OXA23 genes, and the lane 1 indicate the 1 kb DNA ladder, and the lane 2 to lane 11 is the transformants, and the lane 12 is a negative control. So it has been known that the horizontal gene transfer allow the microbial species to acquire new genetic material from outside of their clonal lineage, and this plays a profound role in the accumulation as well as dissemination of antimicrobial resistant genes in bacteria. The horizontal gene transfer of the antimicrobial resistant genes has created the potential for rapid fitness increase in bacteria that are otherwise susceptible to the treatment. Figure 3.4 shows that the transformer with BLA-OXA23 gene inserted for 930 strain plated onto the Luria britannica agar containing ampicillin as well as meropenem with concentration 0.4 microgram per ml. In the middle of the plate, top 10 E. coli was treated to set as negative control. The transformant E. coli that thrive on agar supplemented with meropenem had clarified that BLA-OXA23 was accountable for carbapenem resistance in Acinetobacter baumannii strain 930. Multiple studies had revealed the same findings. In Malaysia, out of 175 carbapenem-resistant A. baumannii, 99% of the isolate contain BLA-OXA23 gene. Meanwhile, out of 99%, 99.4% of the carbapenem-resistant A. baumannii were confirmed carrying OXA23. Meanwhile, in Uganda, BLA-OXA23 gene were the most prevalent carbapenems being detected in all carbapenem-resistant A. baumannii and had been believed to mediate the carbapenem resistance in carbapenem-resistant Acinetobacter baumannii. Meanwhile, in study in Tehran, molecular analysis of beta-lactamase resistant gene in Acinetobacter baumannii isolate showed that 93.33% of the clinical isolate were positive for BLA-OXA gene. Hence, it can be concluded that BLA-OXA23 gene are responsible for carbapenem resistance in Acinetobacter baumannii strain 930. As a conclusion, from this study, we can conclude that Acinetobacter baumannii strain 930 is confirmed to be multidrug resistant with highly resistant towards carbapenems. Secondly, Acinetobacter baumannii also have the ability to acquire antibiotic resistant genes through transformation. And thirdly, carbapenem resistance in Acinetobacter baumannii isolate was mediated by the presence of the BLA-OXA23 genes. And fourthly, is it important to evaluate the effective detection and control measure to control the dissemination of carbapenem-resistant Acinetobacter baumannii isolates within the hospital. These are a few of my references for this study. And that's all from me. Thank you for your time.